Hello, everyone. I am Barbara Alberghini, a PhD student from the Department of Agriculture and Food Sciences of the University of Bologna. And today I'm presenting you my work on specialized metabolites in camelina. Camelina is an oilseed crop gaining increasing attention in recent years. The reasons of the interest in camelina are its adaptability to various environments and growing conditions, and its resistance to pests and diseases. Moreover, camelina has a peculiar oil composition, with an high content of end-3 fatty acids, the presence of tocopherols, and of specialized metabolites. These characteristics make camelina a multi-purpose crop, which can be used for food, feed, and bio-based applications. From studies conducted in recent years, we know that growing conditions can affect the final fatty acid composition of the oil. In particular, temperature during seed filling phase is crucial. In fact, high temperatures can increase linoleic acid contents and oleic acid contents, while low temperatures during this stage can increase linolenic acid contents. However, we don't know much about the influence of growing conditions on other characteristics of camelina oil and seeds. So, the aim of this work is to compare the specialized metabolites content of 6 camelina varieties from different breeding programs grown in 5 consecutive years. The camelina varieties that were tested are imported in table 1, and they were Cypress, Pearl, and 78902, provided by Smart Earth Camelina in Canada, Midas, provided by Agriculture and Agri-Food Canada in Canada, Omega from Poznan University of Life Sciences in Poland, and then WUR from Wageningen University and Research in the Netherlands. Those varieties were tested between 2016 and 2019, as we can see in table 2, at the experimental farm of Bologna University in Cadriano. The experimental design was completely randomized blocks. Fertilization was supplied at the beginning of stem elongation at a rate of 50 kilograms per hectare of nitrogen as urea. All plots were rain-fed, and weed control was manually performed. At harvest, the central portion of each plot, which was 6 meters square, was manually harvested and then threshed. After threshing, representative seed samples were collected from each plot. Seed samples were analyzed by LC-MS Metabolomics at the IJPB in Rennes Institute in France to determine specialized metabolites content as well as specialized metabolite classes. For the extraction, 60 grams of seeds were used, and to each sample, 1 milliliter of methanol, methylterbutyl, and water plus 200 nanograms of apigenin were added. Apigenin was used as internal standard. Metabolomic data were acquired using a UHPLC system coupled to a quadruple time of flight mass spectrometer. Then metabolite accumulation was normalized according to the internal standard, which was apigenin, and the weight of seeds used for the extraction. Going to the results, the ANOVA for agronomic and oil composition characteristics showed significant interaction between year and cultivar for all studied variables, except oil percentage and protein percentage, and you can see in the table. In particular, in 2017 and in 2019, the lowest cumulative precipitation between full flowering and harvest was observed. Moreover, in 2019, the highest max temperatures were registered between full flowering and harvest. These climatic conditions caused a response, in particular from 1,000 kernel weight, with the cypress that has the highest seed weight, as you can see in the first graph, But in 2017 and 2019, the lowest 1,000 kernel weight values for all cultivars were observed. Then in the second graph, we can see the oleic acid content over the 5 years, with a great increase for all cultivars in 2019, with the exception of Midas, which is the pink line, that showed a decrease. Finally, in the third graph, we can see the linoleic acid content over the 5 years, with the maximum values observed in 2019. The only exception is Pearl, which performed best in 2018. As we can see, Pearl is represented by the light green line. Regarding specialized metabolites, the ANOVA showed that total metabolites and specialized metabolites classes were affected more by the year rather than the cultivar. In particular, 10 specialized metabolites classes were identified, as we can see in the table, where also the range values in the 5 years are reported. In the graph, we can see that the maximum accumulation was observed in 2016, while the minimum one was registered in 2019. Correlation study for all cultivar was performed, and for Cypress and 78902, this showed a significant negative correlations between linoleic acid contents and alkaloids, and between linoleic acid contents and amino acids. This could be because Cypress and 78902 have common parental line. Then Cypress also showed a significant negative correlation between linoleic acid content and benzoic acids. Then Pearl and Midas showed significant correlations between seed yield and some classes of standard specialized metabolites. In particular, Pearl showed a significant positive correlations with the cinnamic acids and flavanthiols, while seed yield in Midas had significant negative correlations with flavanones and isoflavones. Finally, in omega, a significant negative correlation between oil percentage and flavanthiols was observed. No significant correlations were observed in WUR, maybe because this is not a very advanced breeding line with the fixed parameters. Then, no significant correlation were observed for glucosinolates. So even if the values of glucosinolates in the years of trials were very changing, this parameter is not related to the other observed. In conclusion, this explorative study further demonstrates the importance of camelina as a multipurpose crop. Moreover, difference in specialized metabolites are mainly attributable to growing condition rather than genotype. So, the study of growing environment and of the relations between oil characteristics and specialized metabolites content could be relevant to predict the presence of specialized metabolites classes in camelina. Thank you very much for your attention.